At each ahu, they placed from 1 to 15 of the huge stone statues. The statues were carved at the quarry at Ranu Raraku. The biggest problem was how to move the statues across the island to their ahu. It seems that the only way this could have been done was by first placing trees along the ground and then sliding the statues across the trees from the quarry to the ahu. This must have taken a large number of trees. Recent scientific work has shown that at the time of the first settlement, Easter Island had a lot of plants and trees. As the population increased, trees would have been cut down to make areas for growing potatoes, wood for heating and cooking, and so on. But the most important need of all was to move the large number of statues across the island. Each clan probably wanted more statues to show its power and status. As they began to compete with one another to build more and more statues, more and more trees were needed. As a result, by 1600, very few trees remained on the island, and the building of statues ended, leaving some statues still at the quarry. Listen carefully. The loss of trees on the island brought about big changes in the everyday life of the islanders. They didn't have trees available for building houses. People began to live in caves and simple reed houses. One question is whether they could have escaped the difficult circumstances in which they now found themselves. Without wood, however, canoes could no longer be built, and only reed boats unsuitable for long trips could be made. And with no wood available, fishing was more difficult because nets had been made from mulberry trees, which had also been used for making cloth. Cutting down the trees badly affected the soil too. Without the trees, the good soil washed away during heavy rains. This made it more difficult to grow food. With less food available, it became impossible to support 7,000 people, and the population fell. After 1600, the once complex society on Easter Island became more and more primitive. Without canoes, the islanders had no means to escape the remote home because of the environmental damage they had caused. Wars started between the clans over the remaining food. Eventually, people began to eat each other. Listen carefully. Why did the Easter Islanders destroy a forest that they needed for their survival? This is a key question. It has been suggested that Easter Island's downfall was not caused by the islanders, but resulted from attacks by neighboring enemy societies or by natural climate changes, such as droughts or the El Niño effect. However, there is no evidence that there were any enemies or friends in contact with Easter Island society after its founding. We also have no evidence at present of big climate changes. It seems most likely that the islanders brought about their own doom through the destruction of their forests and environment. The parallels between Easter Island and our modern world are quite obvious. Because of globalization, international trade, jet planes, and the Internet, all countries on Earth today share resources and affect each other just as did Easter's dozen clans. Easter Island was as isolated in the Pacific Ocean as the Earth is in space. When the Easter Islanders got into difficulties, there was no place to which they could escape. The same is true for us today. But our situation differs in one important way. If the islanders destroyed the environment with just stone tools and their own muscle, the question is what damage we might cause with our powerful tools and machines.
Listen carefully. You've heard about cloning, you've read newspaper articles about cloning, and you've probably seen a science fiction movie or two in which cloning played a factor as well. So, what is a clone? Put simply, it's a copy of another organism that has the exact same DNA as the original. Cloning is nothing new. Nature has been doing it for millions of years. When plants like strawberry plants and potatoes send out runners, new plants grow wherever the runners take root. Each new plant is a clone of the original. Certain animals can clone themselves. A tiny water animal called a hydra can clone itself when a small part separates from the original. That small part grows a new hydra with the exact same genetic material as the original. In special cases, some higher-level animals such as certain insects, worms, lizards, fish, and frogs can develop into adults from unfertilized eggs. Each of the animals that develops has genetic material only from the mother and none from the father. Listen carefully. Most people have heard of the cloned sheep Dolly. 100 years ago, no scientists could have created Dolly. But the first steps in animal cloning didn't start with Dolly. In 1902, the German scientist Hans Spemann used a single hair to divide a two-celled salamander embryo. Each of the two cells developed into a full-grown salamander, and each was a clone of the original embryo. If this process had occurred in nature, we would call them twins. In 1952, two scientists, Robert Briggs and Thomas King, removed the nucleus from the cell of one frog and put it into an egg cell of another frog whose nucleus had been removed. The end result? A cloned frog. The inserted nucleus was taken from an unprogrammed cell of an embryo. Unprogrammed cells of an embryo can turn into any kind of cell to form, for example, bones, blood, a heart, or a kidney. Briggs and King thought that only nuclei from embryos, not adult cells, could be used to create clones. They believed that adult cells, such as skin cells that are programmed to grow new skin over a wound, could only create new cells of the exact same type. Listen carefully. For the general public, it was as if the cloning experiments of Briggs, King, and other scientists had never occurred. Few were reported in the newspapers. But when the sheep named Dolly was cloned, it made front-page headlines all over the world. A mammal had been successfully cloned for the first time, and the implications were clear. If a sheep could be cloned, why not humans? The scientists wanted to genetically engineer sheep and other farm animals so that they could produce proteins in their milk, for example, insulin, that could help humans. The path was not easy. They needed to use adult cells or programmed cells. In some way, they had to trick or reprogram the DNA in the programmed cell to act like an unprogrammed cell. They tried to make a clone 276 times, and it failed each time. On their 277th try, they succeeded. Since Dolly was cloned, scientists have cloned cattle, pigs, cats, and other kinds of animals. This difficult process, however, still has a long way to go. Cloned animals, for instance, are often very unhealthy animals. In February 2003, Dolly was put to sleep after suffering a serious illness. She was only 6 years old. Listen carefully. Many people believe cloning is a technology that can greatly help humankind in the future. Those who support scientific cloning of plants say that it has many benefits for consumers as well as the environment. For example, it can increase food production and create healthier foods. Cloning high-quality plants means that plants for food can be grown more easily, and it can make available crops that are resistant to insects and disease. This can help provide more food for the poor in third world nations. Scientists clone animals not for theoretical purposes. They have real-world practical ends in mind. Gaining that practical end requires the use of bioengineering techniques, not just cloning. For example, scientists hope that someday pigs can be cloned and their organs harvested for human use. But in order for that to happen, bioengineering must be used so that certain genes of the pig are knocked out so that the organs will not be rejected by the human body in which they are implanted. After these genes have been turned off, clones will be made of pigs and their organs used for human implantation. That is the theory, anyway. Listen carefully. Some people worry about the future of cloning. They say it is important that each scientist consider the consequences of his or her work. Those who warn about the dangers of plant cloning, however, don't want to ban cloning. As any gardener knows, plant cloning has been going on for centuries. Rather, they focus on the dangers posed when cloning is mixed with bioengineering. For example, cloning could decrease resistance to disease and insects. If only a very few food crops are grown, and those are clones, then a single disease or insect could destroy huge amounts of crops, causing a famine. If there were more genetic diversity in the crops, they would not be subject to the same disease. Animal cloning can pose dangers as well. One of these is the creation of new diseases. Many of the newest emerging diseases, such as AIDS and SARS, are caused when viruses jump the species barrier. In other words, when a virus originally from an animal passes to man. Some people worry that if organs from cloned animals were transplanted into humans, it could lead to the creation of deadly new diseases.
Listen carefully. My father died suddenly when I was 16. It happened right after I had spent a week alone with him. My mother and brother were away on a trip when he suddenly asked me to take several days off from my part-time job. While I was growing up, he was away most of the time, and he usually just sat in a chair and drank when he was home. Whenever we asked him to tell us about the wars he'd fought in, he would start to cry. World War II and the Korean War had destroyed his spirit. During the Korean War, his best friend covered a grenade with his own body to save my father's life. My dad felt very guilty about that. But the greatest guilt of all involved his family. He apologized to me for not having been a good father. Just as I finally became friends with my dad, I lost him. He had come home from World War II with heart disease and high blood pressure. At the end of that week in 1961, when we finally became closer as father and son, he suffered a heart attack. Listen carefully. A few months